name is Melanie Reiter. I'm a research technologist in the laboratory of Dr. Jennifer Strand. We work in the Division of Cardiology in the Department of Medicine here at the Medical College of Wisconsin. And our lab is very interested in understanding the reasons why the heart fails in patients with muscular dystrophy. So initially, what brought me to this lab was I started out as a graduate student in the IDP program at MCW, and I have a very strong background in microbiology. And through the IDP program, they offer you the opportunity to test out different departments in the graduate school. So I thought I would step outside of my comfort zone and interview with a stem cell researcher, which happened to be Jenny at the time. And after meeting with her and discussing with her the projects that she's interested in, she just came across as a very passionate individual who really cares about her patients and understanding the basic science principles behind why the heart fails in boys with muscular dystrophy. So initially, after meeting her, I was sold. I wanted to join her lab. So the projects that I've been working on since I've started in Dr. Strand's lab focus primarily on understanding the molecular mechanisms that underlie heart failure in boys with muscular dystrophy. And this is a very unique type of heart failure because with respiratory interventions and various types of therapies, these patients are living longer up until their second decade of life when the heart starts to fail. But it doesn't follow the traditional course of more adult type of heart failure. So we want to try and understand exactly how the absence of the dystrophin protein in the membrane of the muscle cell is leading to the enhanced oxidative stress, mitochondrial dysfunction, and elevated cell death that we've seen in our experiments. And some of the experiments that I've done have tried to figure out what is it that causes the heart to fail in these patients. So we've been fortunate enough to retrieve patient specimens from here at Children's Hospital that contain patient-specific mutations. And from there, we reprogram these cells into iPS-derived cardiomyocytes and use them in order to further our understanding of this complex heart failure. So some of the measurements that I've done with my experiments are looking at free radical production, dysfunctions of the mitochondria, specifically mitochondrial membrane potential, and then also measuring cell injury as evidenced by LDH release, and ultimately looking at cell death assays. What we've been finding is that these cardiomyocytes derived from the iPS cells with the mutations is that they die at a quicker rate and they have enhanced levels of oxidative stress and more dysfunctional mitochondria. So then we wanted to try and figure out are there compounds that we could add in order to correct some of these defects. And one of the drugs that we've been testing is called nicorandil, which has been used in Europe for the treatment of angina. And this is a nitric oxide cyclic GMP enhancer. And one of the pathways that is known to be dysregulated in dystrophic cells is nitric oxide signaling. So when we used this drug, we applied it to our cells, we found that it did to a certain extent rescue some of the effects of the oxidative stress in these cells. Really continue to broaden our understanding of the mechanisms of dysregulation of signaling pathways and what else might be causing the heart to fail in these patients. So currently, we have embarked on a new frontier of exploration, and now we're looking at, are the cells secreting something that is either enhancing cell death or possibly rescuing some of the cell death that might be occurring. So we want to try and figure out, are there exosomes that are being secreted, which are small little vacuoles that may contain microRNAs or various other signaling molecules? So are dystrophic cardiomyocytes releasing these? If so, can we capture them and ultimately figure out what's inside and compare this to healthy cardiomyocytes and see if they're also secreting factors? If so, are they secreting more of them, or are they secreting something that may be more beneficial? If so, can we find out a way to enhance that signaling in our dystrophic cardiomyocyte?